Applications of PCR. Here, the PCR can use to study the differences between the genome. Two different organisms genome can study, that is the difference between the two individuals and their genome can easily studied by using of the PCR here. This is the first application. And not only that, here to identification the difference, it is basically useful in the studies of evolution here. Evolutionary studies. In case of the evolutionary studies, so what are the evolutionary studies means? Phylogenetic. It means phylogenetic studies. So the phylogenetic studies, that means the evolution, how it is occurs. The studies into the evolutionary study, that is the studies into the changes into the organism. So one organism to other organism, how they are going to changes, that can easily studied by using of the PCR we are taking. So how we are going to study the evolutionary studies, how we are going to conduct the phylogenetic studies, how we are going to conduct by using of the PCR means, so PCR, a small amount of DNA from any source. So the small amount of the DNA from any source of the organism, it is able to amplify the more the number of the copies by using of the PCR. So that only, so in this PCR technique we are using, so by the taking of tissues or hair or dead organic materials and the dead organic materials of fossil forms here. So fossil forms. So from these sources we are collecting into the DNA material and we are going to amplifying and we are using into the studies of evolutionary aspects here. So like this, the PCR is useful to studying into the difference between the two organisms and the evolutionary studies, that is the phylogenetic studies we are doing. And not only this, can used in forensic medicines here. Forensic medicine. So in this forensic science or the forensic medicine, it is a major technique to constructed into the DNA fingerprinting. DNA fingerprinting here. So in the case of the DNA fingerprinting process, the small amount of the DNA material, so it can be amplified to detecting or to matching into the crime records here. So in case of the forensic medicine or the criminals identification, there they are using into the DNA fingerprint. The DNA, to conducting into the DNA fingerprinting, a small amount of DNA is enough to amplify. So that is going to amplify by using of the PCR. So in the case of this forensic science, that is into the crime scenes we are taking means hair or blood stains or the tissues of the crime. So they are collecting and they are going to amplifying into the number of copies and they are going to study it here. So they are going to observing into the to preparing into the DNA fingerprint. So that is also another advantage or the applications of the PCR value. The fourth one is in gene therapy. Gene therapy. So gene therapy here. DNA amplification methods. Here the desirable genes, whatever they are going to using into the gene therapy. So those genes are commercially we are going to amplifying, we are going to producing into the more number and we are going to using into the therapeutic process of the defected DNA samples here. So there also we are using into the PCR only. So that specific gene which is going to treated into the gene therapy, which is going to use into the gene therapy, that particular DNA molecule or the sample we are going to amplify. So like this, the PCR can use into the number of applications here. These are into the applications of the PCR. Next one we are taking means transgenic animals. Transgenic animals. Earlierly, we are taking the improvement of the genetic characters into the organism. The conventional methods they are following, that is the selection. To improve into the genetic information, genetic characters in animals. So they are doing it the selective breeding methods. Selective breeding process earlierly they are going to conduct. So in the selective breeding methods or the selective breeding process we are taking, so the superior quality or so the desirable quality containing organism they are going to select it and the selectively they are going to breed. So by the number of generations, by following of these selective breeding methods, unwanted characters are going to eliminated from the organism and the desirable characters are these genetical characters are going to introduce into the organism here. So this is into the conventional or the traditional methods here. So before that into the development of the biotechnology and their application. So after the development of the biotechnology application, so biotechnology application, the development of the biotechnology applications, it is very easy to improve into the genetic characters of a desirable organism. That too we are taking the rearing animals. That too, so they are the domesticated animals we are taking. How we are going to improve into the genetic characteristics by using of the biotechnology application in the domesticated animals, means here whatever it is the animal we are going to improve, their genetic characters we are going to improve. So first of all we are selecting and we are introducing into a new DNA fragment into that animal here. So into that animal. So this is generally the process. A new DNA fragments which is not related to that particular animals. It is from the outside of the organism. A new desirable DNA fragment we are introducing into the animal here. So that time this new DNA fragment is established into their genome and they are showing into the expression of this new DNA fragment characters here. So like this, a new stable genetic material we are going to introduce. We are going to forming by using of the biotechnology application. So this is the process is called as a transgenesis. This is called as a transgenesis here. that is transfer of genes. Genesis means the genes. The transfer of genes from one individual to the another individual. The desirable characters we are going to transferring here. This is called as a it is called as a transgenesis. In this process, the transgenesis process, whatever it is an animal, their genome is becomes modifying. That animal is called as a transgenic animal here. So that is called as a transgenic animal. So like this, through the transgenesis process, a new genome or the new DNA fragment we are going to introduce into the another animal to establish into the new genetic character, new stable genetic characters here. So how can we are conducting into the transgenesis process, or how we are preparing into the transgenesis animals we are doing. So the number of steps are involving in the transgenic animals. The process we are taking means, so process we are taking, first one, selection of desired gene. So desirable or the desired, that means whatever it is the genes we want to improve, or whatever the characters we want to improve into that particular animal, that specific genes we are going to select. So by using of the molecular tool techniques, we are separating that particular gene from the selected individuals. So this is called as the selection of the desirable gene. First we are taking into a specific gene from the specific animals. So whatever the animals, they are having the desirable characters, and the selected desirable gene is going to introduce into the transgenic animals. We requires into the some carriers or requires here. So this gene is going to attaching into the vectors. So second one is a selection of the vector. Generally the viruses are generally using as a vectors, phase vectors, like that. The virus we are using as a vector here. So the third step, after selection of the vector, that is a virus, so this desirable gene or gene fragment or genetic material is going to introduce it into the vector, that is a combining of gene into vector. So combining of the gene or introducing of the gene into the vector here, combining with the this selectable or desirable gene fragment into the vector genome. So later on, introducing the vector either the cells, tissues, or adult organism. The introducing of this vector, the cells, tissues, or the adult organism of whatever it is organism we are going to developing their genetic material, that is the transgenic animals, we are going to introducing into the vector here. So later that is the fourth step, and the fifth step we are taking means expression of gene in transgenic animals. So once we are going to introducing the specific genome in the specific organism, that is either their cells or the tissues or the adults. So what is going to happen means that particular genome, whatever we are going to transfer, so that is going to introduce into the transgenic animal. The transgenic animal along with their genetic material, their expression, this newly introduced gene is also going to expressing here. So like this is a process, the number of steps we are going to following to conducting into the transgenesis process. So like this way we are developing into the transgenic animals we are taking. Next one, uses of transgenic animals. So what is the advantage or use of the transgenic animals we are taking? The transgenic animals, it is a powerful tool. Transgenic animals are the powerful tool to study expression and development of organism. So one particular gene and their expression and the development of the organism is very useful to studying these characters. The transgenic animals are very useful tools here. So this is the first advantage or the use of the transgenic animals here. And the second one, transgenic animals. So transgenic animals act as good models. So good models for different disease research here. So different types of the diseases, to study into the different types of the diseases and their causing genomes. So there we are not going to utilizing into the, we are not going to conduct the experiments on the humans. So there we are going to using into these transgenic animals to recognizing into the different diseases and their treatment process. We are going to using into the transgenic animals. Example we are taking is sickle cell anemia, SCA, Alzheimer's, so cancer, so etc. So these are the different diseases. To recognizing into these diseases and to search into the drugs or to conducting in the experiments on these disease causing pathogens and their genetic material, the transgenic animals are going to be utilizing as a good models here. This is the second advantage or the use here. And the third one is transgenic animals. So transgenic animals are used in therapeutic proteins production. So therapeutic proteins, transgenic animals are used into the pharmaceutical industry to producing into the therapeutic proteins. That means the specific protein molecule which are using into the treatment of diagnosis of some specific diseases. Those proteins are therapeutic proteins are going to producing by using of the transgenic animals here. So this is another advantage here. And the fourth one, toxic substances. The toxic substance toxicity. The toxicity in the toxic substances. So toxic substance toxicity in animals. So in animals, to conduct to identifying the toxicity into the toxic substances in the animals, we are using into the transgenic animals here. Transgenic animals we are going to use here. How we are going to identifying the toxicity means the non-transgenic animals and the transgenic animals. We are taking into the transgenic animals and the non-transgenic animals. We are taking these two. we are injecting this toxic substance in the both the animals. The effect of the toxic substance into the transgenic animal and the effect of the toxic substance in the non-transgenic animals, we are going to study here, comparing here. So here based on that, we are going to study the toxicity of the transgenic animals here. So this is another advantage here or use here. So that is a toxicity. And producing of transgenic animals are produced industrial important food materials. So important products. So food materials and the different types of the products we are going to use by using of the transgenic animals. This is another application. And lastly, we are taking the transgenic animals are used to improve the quality and quantity. So quality and the quantity of food materials in industrial scale. So this is also another advantage or the use here. And we are taking the another use of the transgenic animals, that is vaccine studies. So here the effect of the vaccine, the preparation of the vaccine. So compared to the different types of the to studying the different types of the animals, vaccine studies also transgenic animals. So like mice and the pigs we are going to using here. So these are the different types of the advantages of the uses of the transgenic animals we are going to take. So next one is the biological products and their uses here. So what is meant by the biological products? The products so which are produced by using of animals or the cells. So the products which are produced by using of the animals here. So before that you are taking either the plants or the animals traditional methods, so the different products we are going to getting here. But here by using of biotechnology applications of biotechnology. So by the development of the biotechnology, so the products, the industrial scale or the industrial range, the certain type of the products we are going to using. So by using of the different types of the animals. So these products are called biological products here. Generally, what are the products? They are comes under the biological products means. So they are the proteins. That means they're therapeutic proteins, antibodies, so hormones, enzymes, so etc. So these are the different types of the products we are going to produce in the industrial scale by using of the animals here. Generally, different types of the products we are going to producing by the industrial application. Normal also the industry we are going to use or we are going to producing the different types of the products here. Why we are going to using here the animals? Animals are acting as a here in this process of the biological production. Animals act as bioreactors. So the meaning of the bioreactor, we know that. So bioreactor is nothing but as the animal itself. Bioreactor is nothing but as the biological products giving the structure is called as the tool which is going to produce into the products that is comes under the bioreactors. Industrially, in the synthetic synthesis process, we are taking a larger tanks or the steel tanks we are going to using as a bioreactors. There, that is in the industrial application. Here, what we are going to using to producing the specific proteins, like that as a protein, therapeutic proteins, antibodies, hormones or the enzymes, we are using the animals. Animals act as a bioreactor here. So like this, by using of the animals, so we are going to producing the some specific kind of the products. This is a process called as a biological production, the biological products here. So in this process, what we are going to do means the specific gene. So biotechnology application is nothing but as a specific gene we are going to introduce it into the animals. So introduce it into the animals. So in their cells or tissues or total organism, we are using into the either the cells or the tissues or the total organism or their organs we are using. So by using of either cells, tissues or the total organism, so introduced into a new genome. So in the animal, and this genome is going to expressing and producing a new products here. A new product is going to produce. So this is the process we are going to conducting into the biological products formation we are taking. So already we discussed that is the transgenic animals. The transgenic animals, what is going to occur means that is a process is nothing but as a biological production only. So there, for example, we are taking monoclonal antibodies. Monoclonal antibodies. So monoclonal antibodies are blood proteins. So like that as a antiseras or hormones. So that too human hormones, like that as a growth hormone, GH, human growth hormone, and enzymes. So like that as a insulin, human insulin. So these are the different types of the products we are going to producing by using of the animals. A new products are going to producing. So by using the animals, these are called as a biological products here. So why we are going to use the animals as a bioreactor? Why we are not following the industrial methods to producing into the different products? Means here, in case of the organism or the animals or the humans we are taking, means biological proteins. So generally the biological proteins or the biological products. So biological products not recognized, the immune system you are taking, the immune system is not recognizing into this biological product as a non-self here. So normally from outside to any one of the product we are going to insect it into the organism, it detect or it is recognized, the immune system is going to detect it is a non-self. But in this case, the biological products or the biological proteins, whatever we are going to producing. So monoclonal antibodies or the hormones or the insulin, enzymes, whatever it may be. So those are not going to recognized as a non-self materials here. So that only these biological products we are going to using into the different types of the disease treatments here. They are the cardiac disease and graft resections, so graft resection and cancer treatment and insulin deficiency So in these all different types of the diseases we are using into the biological. See here what it is happened, whatever it is the products we are going to produce, for example the graft resection process. So sometimes by the transmission of the different organ transplantation, some tissues are going to resected here. Why? They are act as a non-selfing materials. To reducing into the this graft rejecting reactions, so to suppressing into the immune reactions, we are going to using into the biological products, we are the therapeutic proteins are going to using here. So there it is going to reducing the graft rejection reaction. Or into the cardiac diseases, so some of the cardiac diseases, that is the their walls, the repairing of the walls, cardiac walls, and even though cancer treatments or the insulin deficiency. In case of the human insulin deficiency, there also artificially or recombined rDNA technology. By using of the rDNA technology, we are going to producing into these biological products. So these biological products are using to treating into these different kinds of the diseases. So this is the advantage of the biological products. So that we are going to producing by using of the animals at different types of the different categories of the products are we are going to using here, producing here. So this is regarding to the biological products. So today's organism and the population. Here this organism and the population in the chapter ecosystem or ecology. Ecology. So what is mean by the ecology means the study of ecosystems. What is mean by the ecos? Here the word ecology is derived from the word oikos here. Oikos means house. Ecology means study. So ecology means studies. House studies. That means the organism where they are going to living, where they are going to surviving. So that particular region and particular region. So the studies of those regions and the organism with their surroundings, the relationship is going to study into the ecology here. So this is a major aim of the ecology. The studies of the organism and their surroundings, how they are going to related, how they are going to interact with their surroundings, that is going to studied into the ecology here. In this ecology we are taking means, so ecological studies we are going to discussing into these words, that is the population, community and ecosystem and the biome and the biosphere. So one by one we can go through these all the words. So first one is the population. Population is nothing but as a group of organisms. A group of organisms, one particular region. So a number of organisms together, they are living in the one particular region. So that group of the organism or individuals is called as a population here. Okay, that group of the individuals are called as a population. And the community. So community is nothing but as a number of populations. Here, number of populations. So that means a different number of population. They are not belongs to the same. So a different types of population, different number, not types, a different number of populations. Together, they are living into the one particular region. That is called as a community. And the ecosystem. Ecosystem, it comes the number of communities with their environment. So the relationship between into the communities, that is the organism and with their environment. That means biotic and the abiotic factors. Biotic and abiotic factor. The relationship between into the biotic and the abiotic factors of a certain area in a certain time, it is going to explained by the ecosystem. It is called as an ecosystem. Like that as a biome. Biome is nothing but as a number of ecosystem, their distribution in the geographically, that studies is called as a biome. That means one particular region, one particular continent we are taking, means there the number of ecosystems are going to distributed. So their relationship is, those are all together we are calling as a biome here. And the biosphere. So biosphere is nothing but as a earth and its biotic, abiotic. So factors together we are called as a biosphere here. So these are the basic words we are going to using into the studies of the biotechnology here. Next we are going to take organism and environment. Organism and environment here. Organism and the environment. In any organism, in environment or in the ecosystem we are taking, means they are going to showing the adaptations here. Adaptations. So adaptations here. Adaptations are nothing but as a changes in organism towards the environment. So here environment is continuously going to change. So according to those changes, the organism, to survive the organism beneficially, so to withstand that particular changes into the environment, the organisms are going to change in their morphologically or the physiologically. That means their behavioral changes are occurs, their morphological changes, their structural changes are occurs, their functional changes are occurs. Those changes are called as adaptations. So organisms are going to adapt it. They are going to adapt it according to the changes which are occurs into the environment. So like these organisms are adapting to surviving into a particular environment, and these are having the relationship with the environmental changes. According to the environmental changes, their body, so changes also going to conducted here. So this is the regarding to the how the organisms are going. So how organisms are influencing by the changes of the environment we are taking. Next one is the habitat. Habitat here. So habitat is nothing but as a organism where it is surviving, organism where it is living, so a particular area. So organisms are where they are going to living, one particular region, one particular area. That area is called as the habitat of the organism. The habitat of the organism here. And niche. or it is also called as a biological niche. So biological niche. So what is mean by the biological niche, or the niche means here habitat we are taking, organisms are going to survive in the one particular area. This area can share into the so many number of the organisms. One particular region is called their habitat. So where the organisms are going to survive, that particular entire area is called as their habitat here. So it may be sharing by the some other organisms also. But in the niche or the biological niche means it is a specific unit of area. So the specific unit of the place or into the area, so which is present into their habitat or the particular organism. That means it is a specific addressing unit of the one particular individual. So one habitat can share into the so many number of the organism, but the niche is specific for the each organism here. How can we are going to expand this one means, for example, the habitat is nothing but as a where we are going to live in. So for example, anybody they are going to ask me, sir, where are you from, like that they are asking means just we are telling into the our village name. So that is into the habitat, suppose. So niche, so are the biological niche. Suppose anybody they are asking within the same village, suppose anybody they are going to ask him, so where are you from or where is your house, means that time we are explaining into the specific address, that is into the street, so house number and the particular region. So that is belongs to the only for us, that particular specific unit of the area is called as a niche here. So like this, the habitat and the niche, so these are into the specific for the each and the every organism. So niche is a specific for the unique for the one individual. So habitat is a common for the a few number of the organisms here. So like this we are taking the ecological studies. These are the basic terminologies or we are going to using here.